Okay, in this video, we're going to look at the structure of antibodies, their use as monoclonal antibodies, and then HIV. So here is the structure of an antibody. Important thing to know is that it is a quaternary protein, and that's because it contains more than one polypeptide chain. So you can see we have 4 here. We've got 2 heavy ones, which are the black ones shown here, and 2 light chains. It's made up of a variable region, which changes in each antibody, and a constant region, which is the same in all of them. The antigen binding site, which is specific and complementary to the antigen, is found at the top of the variable region. We then have disulfide bonds or bridges, which hold the structure together, and then a hinge region, which allows the antibody to open and close, as shown here. We've got an open antibody and then a slightly closed one. So when we're describing antibodies binding to the antigen, it's important we talk about it binding to a specific and complementary antigen, and that the antigen binding site is on the variable region, forming what we call an antigen-antibody complex. Now, antibodies have 2 main functions we're going to look at. The first one is neutralization. Neutralization is the idea that it binds to the pathogen, or in the case of antitoxin, to the toxin, and prevent the pathogen or toxin causing damage. And then agglutination, which is the idea that the pathogens are stuck together, they're clumped together by the antibodies, as shown in the picture below, and therefore more likely to undergo phagocytosis. Monoclonal antibodies, as the name suggests, are antibodies produced from a clone of plasma cells. So one type of plasma cell producing one type of antibody. And there are 2 main uses. The first use is in medical diagnosis. In other words, for seeing whether someone is a carrier of an antigen, for example, so maybe they're HIV positive, or they've got the COVID antigen in their respiratory system, or seeing whether someone has antibodies for a particular antigen is the other possibility. So if you think about looking at what our antibody titer is at, that's the sort of thing that we're looking for. The second use is cancer treatment. We know that cancer cells have a specific antigen on their surface, which is unique to cancer cells. So that's shown as the red blobs on the surface here. And if we can make antibodies, and those antibodies are really specific and combined to the antigen, and they bring with it a drug that's attached to them, then that drug will only kill cancerous cells. It's targeted and personalized chemotherapy. Obviously has a lot less side effects because it's damage healthy cells, but by nature it's going to be expensive. So we're going to think about how monoclonal antibodies are used in what we call the ELISA test. And there's 2 types of ELISA tests. The first one we're doing is looking for the antigen. The description is on the right-hand side. I'm going to actually sort of draw it out. So the first thing we do is we coat our plate with the antibody for our specific antigen. So for example, maybe we're looking to see if someone has the HIV virus in their bloodstream. So we coat with an antibody which is specific for HIV. Now, when we coat the plate, some of the antibody may not stick, so we need to wash it afterwards just to remove any antibodies that haven't stuck. We can then pour on the sample like so. In this particular case, I'm going to assume my person has the HIV, so the antigen is going to bind to my antibody. Again, some of it will just be sitting around and not stuck, so we're going to need to wash it to remove any that aren't. Put my tray back in again. And then the next thing we're going to do is pour on another antibody, a second antibody, but this antibody is a little bit special because it contains, attached to the end of it, an enzyme. And if the antigen is present, then the antibody will bind. Again, some might not bind, so we have to wash it. And then finally, we pour on a substrate, and what the substrate does is the substrate changes color. So for example, it goes from orange to blue. Okay, so my substrate goes blue. So I pour on an orange substrate and the enzyme changes its color to blue. So my sample goes blue and I know they had the antigen present. Let's look what happens if they don't have it present. So we start off exactly the same. Coat the first one with antibody, then wash it. But this time when we pour on the sample, there's nothing there, nothing to stick. So therefore, when we pour on the second antibody, it doesn't actually stick to anything, which is why it's now really important that we wash it away, because this time when we pour on the substrate, it will just stay orange. There's no enzyme to convert it to blue. The other possibility is that you're looking for the antibody. The principle is much the same, but we just do it in reverse. So this time we're going to coat the bottom of the tray with the antigen. Again, I'm going to wash it to prevent any unbound antigen staying behind. And I'm going to pour on my sample. Now I'm looking for the presence of an antibody. So I might be looking to see whether they have any HIV antibodies or they have COVID antibodies, whatever it is. And if the antibody is present, again, it will stick to the antigen like that. It will bind to it. Again, I wash it to remove any unbound ones. And then I pour on my second antibody. My second antibody is complementary to my first antibody, so it can bind to the bottom of it like this. And my second antibody, if you remember, has the enzyme attached like so. And then I can pour on the substrate and see if it changes color or not. But so it's exactly the same principle. I still need a second antibody. What's different is the order in which the bottom two are put in. Okay, so the last thing for us to think about is HIV. Now, HIV is a virus. Viruses, we know from learning earlier in the course, are not cells, and they're made up of nucleic acids inside a protein coat. So that's the same. So we've got RNA in the case of HIV. It's what we call a retrovirus. And it is encased in this capsid, this protein coat, but it also has the enzyme reverse transcriptase. The job of reverse transcriptase is to take RNA and convert it into DNA. It then has a viral envelope, which is basically the same phospholipid bilayer as a cell surface membrane, and then glycoproteins, which are attachment proteins. So if you're asked to describe an HIV, you would start literally by saying that it has RNA and reverse transcriptase within a capsid, and then it's surrounded by a lipid envelope, lipid viral envelope, studded with attachment glycoproteins. So we know that HIV infects our T helper cells, so it lives within our immune system. So if we have a look at how it works, we have a cell like so, and the cells have various proteins on their surface naturally. So this is a T helper cell. We have the nucleus inside with its DNA. Just draw it like so. Not going to draw all 46 chromosomes. And then we have the virus, the HIV, okay, which looks like that with its attachment proteins and its capsid and its RNA and its reverse transcriptase. And what they found is that the attachment proteins can attach to proteins on the surface of the T helper cell, and that allows the lipid envelope to fuse with the envelope of the cell itself and release into the cell RNA and reverse transcriptase. Reverse transcriptase will convert the RNA into DNA, and that DNA will then insert itself into the human genome. And the human genome will now transcribe and translate viral proteins, so more reverse transcriptase, more capsid proteins, plus make the RNA, so that eventually we can construct more capsids, which will then bud out of the T helper cell, forming new HIV particles that can go infect other cells. Now, initially, you might just get a few budding off. So one buds off, goes, infects another cell, and so on. But there comes a point at which the replication is really, really fast, and it starts to destroy the T helper cells. And once the T helper cells are getting destroyed in large amounts, the person's immune system becomes very, very, very low, and we say that they have AIDS. What does it mean if they have AIDS? It means they don't have many T helper cells, and therefore they are prone to opportunistic infections, which are other infections that will kill them. And it's because of the fact that the T helper cells have become infected and then destroyed. We can treat people who are HIV positive with antiviral drugs to try and prevent HIV from reproducing, and they are quite effective, but at some point in their life, they will start to reproduce.